Right, good evening. I'm back with you. I'm Bobby here. Now we're moving on to the next chapter of Kingdom Monera. Right now, Monera are a group of all prokaryotes. They're basically unicellular, maybe mycelial, colonial, and filamentous. Cell wall is made up of peptidoglycan, polysaccharides, and cellulose. They do not contain any organized nucleus with distinct membrane. DNA is naked. That is not associated with histone proteins. It is called nucleoid. All the membrane-bound cell organelles are absent, like mitochondria, lysosomes, Golgi bodies, plastids, etc. In autotrophic forms, thylakoids are present without chloroplasts. Spindle apparatus do not develop at the time of cell division. Single-stranded flagella composed of flagellin is present. This is a flow diagram. Bacteria based on shape: Coccus, Bacillus, Vibrio, Spirullum, and we get to an explanation after I finish up this portion down here. And they give rise to Diplobacillus, Palisade Bacillus, Streptobacillus, further on to Micro, Diplo, Tetracoccus, Streptococcus, Staphylococcus, Succinate, and bacteria based on flagellation. The one above is bacteria based on shape, this one. Right? Those are based on flagellation will be Atricus, Monotrichus, Lophotrichus, Amphitrichus, and Peritrichus. All right? Just going back to the subject proper here. Ribosomes are 70S type, that is subunit 30S and not 50S. Reproduction is by binary fission or budding. They have various modes of nutrition like saprobic, parasitic, chemoautotrophic, photoautotrophic, and symbiotic. Right? Some are capable of nitrogen fixation. Monera occur in all environment. Example, Archaebacteria live in extreme climates. The kingdom includes all prokaryotes like bacteria, Actinomycetes, mycoplasma, and Cyanobacteria. Bacteria are the smallest of free-living organisms, mostly unicellular. Bacteria possess various forms and shapes and are 4 different types: Coccus, Bacillus, Vibrio, and Spirullum. This is the flow diagram I've been speaking to you so far. And we move on to the next page. That being said, bacteria possess a distinct cell wall with different wall layers like capsule or slime layer, plasma, membranes, flagella, and pili. The slime layer The slime layer or capsule is made up of polysaccharides and amino acids and act as osmotic barrier. Cell wall is made up of polysaccharides, proteins, and lipids, and peptidoglycans. Right? Or mucin. Sorry, a spelling correction. Let me just correct it for you. Murine. Right? Plasma membrane is tripartite in nature. Mesosomes, a simple enfolding of plasma membrane containing respiratory enzymes like oxidase and dehydrogenase. Right? The plasma membrane is tripartite in nature. Mesosomes, a simple enfolding of plasma membrane containing respiratory enzymes like oxidases and dehydrogenases. Flagella is made up of flagellin. Pili are small hair-like outgrowth, present on bacterial cell wall made up of pilin protein. Pilin helps in formation of conjugation tube and agglutination. Based on the nature of staining, bacteria may be gram-positive retains the blue stain, or gram-negative does not retain the blue stain. Gram staining, heat-fixed smear of bacteria. Crystal violet, rinse with water. Right? And gram staining, heat-fixed smear of bacteria, that's the process, okay? Crystal violet, rinse with water, add dilute iodine solution, all bacterial cell appears a deep blue, treat with 95% alcohol. Right? And now, once it's treated with 95% alcohol, cells retain purple color. Right, and inner to the wall layers there is present matrix or protoplasm, which includes nucleoid, plasma episomes, ribosomes, and granules. In the center of the bacterial cell, there is present nuclear material DNA without any nuclear membrane naked. DNA in bacteria is double helical and circular. This incipient nucleus or primitive nucleus is named as nucleoid or genophore, sometimes called single naked chromosomes. Besides this nuclear DNA, there is some extranuclear or extrachromosomal DNA, which is known as plasmid. And on this table, difference in the cell walls of gram-positive and gram-negative bacteria. Gram-positive bacteria and gram-negative bacteria. Cell wall is more thick, and in the other case, cell wall is thin. Thickness varies from 10 to 15 nanometers and 25 to 30 nanometers. Cell wall is a 3-layered homogeneous layer. 3. High content of less content of peptidoglycan, 20 to 80%, peptidoglycan, 10 to 20% of the dry weight of the cell. Teichoic acids may be present, and the other teichoid acids will be absent. Very little lipid content, 0 to 2%, and high lipid content, 10 to 20%. Only a few amino acids are associated with the muramic acid complex. Large variety of amino acids is associated. Lipopolysaccharide layer, LPS, is absent, the other it is present. Periplasmic space is absent, and the other it is present. The plasmid are small, circular, double-stranded DNA molecules that are separate from main bacterial chromosomes. And replicate independently. The term plasmid was given by Lederberg in 1952. Plasmids have an independent existence. Plasmids carry genes for fertility, antibiotic resistance, R factor, and bacterium collagen production is cholecynogenic factor. F factor or fertility factor is responsible for transfer of genetic material. R factor or resistance factor provides resistance against drugs. Cholecynogenic factor produces cholecyns, which will kill other bacteria other than which produces the cholecyns. The term episome is applied to extranuclear genetic material which may remain integrated or free state. Example, F factor, temperate, phage, etc. Ribosomes are evenly distributed in the matrix. Ribosomes are 70 S type, 50 S plus 30 S. Ribosomes are the seat of protein synthesis and are made up of RNA and the protein. Okay, going to the next page. There are present different types of granules like volutin granules, fatty acid granules, lipid granules, glycogen, and sulfur granules. According to the mode of respiration, bacteria can be aerobic or anaerobic. Each of them is further of two types, obligate and facultative. Obligate aerobes are bacteria which can respire only aerobically. They generally get killed under anaerobic mode of respiration, example Bacillus subtilis. Facultative aerobes are bacteria which respire anaerobically under normal conditions but can respire aerobically when oxygen is available. Most of the photosynthetic bacteria belong to this group. Obligate anaerobes are bacteria that respire only anaerobically. They generally get killed under aerobic conditions, for example Clostridium botulinum. Facultative anaerobes are bacteria which generally respire only aerobically, but switch over to anaerobic mode of respiration if oxygen becomes deficient. Nutrition. Bacteria show both autotrophic and heterotrophic nutrition. Autotrophic nutrition consists of manufacture of organic materials from inorganic raw materials with the help of energy obtained from outside sources. It is of two types, chemosynthesis and photosynthesis. The bacteria possess photosynthetic pigments of two types, bacteriochlorophyll and bacteriophycophytin. Chlorobium chlorophyll. The two types of pigments respectively occur in purple bacteria, example Thiopedia rosea and Rhodopseudomonas, in membranes of thylakoids. No oxygen is evolved in bacterial photosynthesis. Such type of photosynthesis is known as inoxygenic photosynthesis. Water is not used as a source of reducing power. Instead, hydrogen is obtained either directly, some purple bacteria, or from various types of inorganic and organic compounds, for example, hydrogen sulfide, green bacteria, aliphatic compounds, purple non-sulfur bacteria. The Autotrophic bacteria are bacteria which are able to manufacture their organic food from inorganic raw materials with the help of energy derived from exergonic chemical reactions involving oxidation of organic substance present in the external medium. They are of various types. Such as the nitrifying bacteria, Nitrosomonas and Nitrosococcus, obtain energy by oxidizing ammonia to nitrite. Ammonia plus oxygen gives NO2 minus plus 2H2O plus energy. Nitrosides and nitrobacteria oxidize nitrites to nitrates, so 2NO2 minus plus O2 gives 2NO3 minus plus energy. Sulfur oxidizing bacteria, Bacillkys, a colorless sulfur bacterium, oxidizes hydrogen sulfide to sulfur in order to obtain energy for chemosynthesis. 2H2S plus O2 in the presence of Bacillkys, 2S plus H2O plus energy is released. Thiobacillus thioxidans, another sulfur bacterium, oxidizes sulfur to sulfate state. 2S plus 2H2O plus 3O2 gives Bacillkys and thioxidants. 2H2SO4 2H2SO4 plus energy. Iron bacteria, Ferrobacillus ferrooxidans, obtains energy by oxidizing ferrous compounds to ferric forms. And 4FeCO3 plus 6H2O plus O2 gives 4FeOH3 plus 4CO2 plus energy. Other chemosynthesis bacterium, Methanogens, oxidizes methane into carbon dioxide and H2O. CH2 plus 2O2 gives CO2 plus 2H2 plus energy. Heterotrophic bacteria may be saprophytic, parasitic, or symbiotic. Saprophytic are living bacteria which obtain their food from organic remains, example corpses, animal excreta, fallen leaves, vegetables, fruits, meat, jam, jelly, bread, and other products of plant and animal origin. Symbiotic bacteria live in mutually beneficial association with the organisms. Enteric bacterium, Escherichia coli, live as a symbiont in human intestine. Parasitic bacteria live in contact with the other living beings for obtaining nourishment or special organic compounds required for growth factors. Bacteria show four major phases of growth in a fresh nutrient-rich medium: lag phase, log phase, logarithmic or exponential phase, stationary phase, and decline phase, death phase. These phases constitute the standard bacterial growth curve. Reproduction. Bacteria shows three methods of reproduction: vegetation reproduction, asexual reproduction, and sexual reproduction. We have vegetative reproduction, which includes budding and binary fission. Binary fission takes place during favorable conditions. The bacterial cell divides by a constriction into two halves. The nuclear cell divides by a constriction into two halves. Asexual reproduction takes place by endospore formation, conidia, and zoospores. During unfavorable condition, highly resistant single spore is formed inside the bacterial cell, which is known as endospore. Endo means inside or within plus spore. Endospores have a characteristic structure that is have an outer thin exporium followed by one or many layered spore cord, followed by many concentric layers of cortex, which is followed by cell wall, cell membrane, and matrix. The endospore is highly resistant to very high and very low temperature, strong chemicals and acids due to calcium, dipicolonic acid, and peptidoglycan in cortex. Dipicolonic acid, DPA, helps in stabilizing its proteins. DPA and calcium ions provide resistance to heat when favorable conditions. When favorable conditions come out, outer layers rupture and active bacterial cells come out. So this is a method of perination. That is the tide of unfavorable conditions, and some people say it's reproduction without multiplication. Sexual reproduction occurs in the form of genetic recombination. There are three main methods of genetic recombination: transformation, transduction, and conjugation. Transformation. Here, genetic material of one bacterial cell goes into another cell, bacterial cell, by some unknown mechanism, and it converts one type of bacterium into another type, non-capsulated to capsulated form. This was first studied by Griffith in 1928. And Streptococcus and Diplococcus pneumoniae, and hence is known as Griffith effect. Transduction. In this method, genetic material of one bacterial cell goes to another bacterial cell by agency of bacteriophages or phages, viruses infecting bacteria. Transduction was first of all reported in Salmonella typhimurium by Zinder and Lederberg in 1952. In abortive transduction, the new gene does not integrate with the recipient genome and is lost. Conjugation. Cell-to-cell union occurs between two bacterial cells and genetic material. DNA of one cell, bacterial cell, goes to another cell lengthwise through conjugation tube, which is formed by sex pili. Conjugation was first reported by Lederberg and Tatum, 1946, in E. coli bacteria. Conjugation occurs between donor cell and recipient cell. Donor cell is having sex pili and F factor, whereas recipient cell is having both. In donor cell, factor F may unite with the main genome or nuclei DNA, and this donor cell is called HFR donor cell, high frequency donor cell, and here transfer of DNA is rapid. Importance of bacteria. Useful activities of the bacteria. Role of bacteria in agriculture is increasing soil fertility. Some free-living nitrogen-fixing bacteria like Azobacter and Clostridium have the capacity of fixing atmospheric nitrogen into nitrogenous substances, hence increases soil fertility. Similarly, symbiotic bacteria, Rhizobium, all fix atmospheric nitrogen. Nitrogen monoids converts ammonia to nitrates, which is further converted into nitrates by nitrobacter's nitrification. In dairy industry, lactic acid. In the dairy industry, lactic acid, bacteria, that is Bacterium lactic acidi and Bacterium acidilactici, convert lactose of milk into lactic acid and hence milk turns sour. In other industries, in vinegar industry, Azotobacter acetic converts sugar solution into acetic acid. Butyl alcohol and acetone are produced by activity of Clostridium acetobutyrrhizium. Retting or separation of fibers from stalks, example jute, sunhemp, linum, flax, etc., is done by water-inhibiting bacteria, Clostridium butyricum. Flavoring of tobacco leaves is done by Bacillus megatherium. Curing of leaves of tea is done by Mycococcus condensans. So we have a lot of explanations there. The role of bacteria in sewage disposal. For the breakdown of sewage in simple substances, bacteria and algae play important roles. Sewage oxidation ponds or tanks have luxuriant growth of bacteria and algae. Role of bacteria in petroleum pollution. Petroleum pollution in water bodies is checked up to some extent by Pseudomonas. Role of bacterium in human beings. E. coli, gram-negative bacteria live in colon region of intestine of man and other animals and play an important role in digestion process. Many antibiotics are produced from bacteria. Antibiotics are substances produced by microorganisms which in low concentration are antagonistic to the growth of other microorganisms. Medicinally, antibiotics are those organic secretions which check the growth of different pathogens without harming the host. Table: a role of bacteria in medicine. Antibiotics and bacteria from which obtained. That is Subtilin is Bacillus subtilis. Polymyxin is Bacillus polymyxa. Streptomycin and Streptomyces grisens is cyclohexamide. Chloromycetin is Streptomyces venezuelae, a chloramphenicol. Tetramycin is Streptomycin rhimosus. Oromycin is S. aurifaciens. Erythromycin is S. erythraeus, and neomycin is S. fradiae. Harmful activities: spoilage of food. Saprophytic bacteria causes rotting of vegetables, fruits, meat, bread, souring of milk, cheese, butter, spoilage of jams, jellies, and pickles. Destruction of penicillin by Bacillus brevis. Cotton spoilage is done by bacteria called Clostridium botulinum. Desulfurification of soil. Soil sulfate is changed to hydrogen sulfide by desulfuricants. Let me just read that. Desulfuricants. All right, okay. And next is about reduction of soil fertility. There are some denitrifying bacteria in soil which can convert nitrates into free nitrogen. Denitrification, example, Bacillus denitrificans, Micrococcus denitrificans, and Theobacillus denitrificans. Table: bacterial diseases of plants, animals, and humans. Name of disease and the causal organisms. Human beings. Pneumonia is Diplococcus pneumoniae. Typhoid is Salmonella typhosa. Cholera is Vibrio cholerae. Plague is due to Pasteurella pestis. Meningitis is due to Neisseria meningitidis. Gonorrhea is Neisseria gonorrhea. Syphilis is by Treponema pallidum. Diarrhea by Bacillus coli. Gastroenteritis by E. coli. Diphtheria by Conibacterium diphtheriae. The tuberculosis is by Mycobacterium tuberculosis. Gangrene is by Clostridium perfringens. Jaundice is Leptospira. Icterohemorrhage. Whooping cough is by Haemophilus pertussis or Bordetella pertussis. Tetanus lockjaw is Clostridium tetani. Bacterial dysentery is Shigella dysentery. Leprosy is Mycobacterium leprae. Animals, anthrax caused by Bacillus anthracis. Blackleg disease caused by Clostridium shawai. Plants, soft rotted potato, Pseudomonas solanacearum. Citrus canker, Xanthomonas citri. Bacterial blight of paddy is Xanthomonas oryzae. Tinder disease in wheat, Corynebacterium tritici. Potato wilt is caused by Pseudomonas solanacearum. Fire blight of apple and peach is Oventa amylivora. Apple and peach, crown gall. Fire blight of apple and peach is called Ovenia amylivora. Crown gall of beet sugar is caused by Acrobacterium, and black rot of cabbage is caused by Xanthomonas campestris, right? The spirochetes, some of the types of bacteria. Spirochetes are free inhabitants of mud and water and are heterotrophic bacteria. Many diseases are caused by them, as Treponema pallidum causes syphilis. Leptospira causes infectious jaundice, and Borrelia causes relapsing fever. Besides, some spirochetes are found in teeth. Rickettsia are gram-negative obligate pleomorphic but walled intracellular parasites which are resident of or are transmissible from arthropods. They are intermediate between true bacteria and viruses. Chlamydia are gram-negative intracellular parasites of about 0.24 micrometer size, often grouped along Rickettsia, but differ from them in reproductive cycle that involves formation of initial or reticulate bodies, RB, and elementary bodies inside host phagosome. Chlamydia trachomatis causes conjunctivitis. Sexually transmitted non-gonococcal urethritis, epididymitis, cervicitis, proctitis, and lymphogranuloma venereum. C. pneumoniae causes pneumonia and bronchopneumonia. Actinomycetes. Actinomycetes are mycelial aseptate branched filaments. Bacteria which form radiating colonies in culture. Because of this, actinomycetes are formerly called ray fungi. Mycelial form is reduced in Mycobacterium and Corynebacterium. Mycelia have a diameter of 1 millimeter or less. Walk contains mycolic acid, fatty acid, lipid, and walks. Different modes of reproduction are by conidia, sporangiophores, astrospores, or oedia, and fragmentation. Most of the actinomycetes are separotrophic and constitute an important component of decomposers, for example, Actinomyces, Streptomyces. A few are pathogenic in plants, animals, and humans, example, Mycobacterium. In pathogens, Actinomyces or Mycobacterium, a derivative of mycolic acid called mycoside or cord factor, is involved in causing diseases. A number of antibiotics are produced by Actinomyces, especially the genus Streptomyces, streptomycin, chloramphenicol, tetracycline, terramycin, erythromycin, viamycin, novapicin, and nystatin. The term antibiotic was given by Abraham Selma Wachsman in 1888, who lived from 1888 to 1973, who also extracted the first antibiotic from Streptomyces griseus and named it streptomycin. First, Wachsman was given Nobel Prize in 1952. Now, Archaebacteria are characterized by absence of peptidoglycan in their wall. Instead, the cell wall contains protein and non-cellulose polysaccharides. It has pseudomycin in some methanogens. The cell membranes are characterized by the presence of a monolayer of branched chain lipids. Archaebacteria even now live under extremely hostile conditions where very few other organisms can dare subsist. Example, salt pans, salt marshes, hot sulfur springs. Their RNA nucleotides are quite different from those of other organisms. The Archaebacteria are of two broad categories: obligate anaerobes and facultative anaerobes or anaerobes. Archaebacteria are of three types: Methanogens, halophiles, and Thermoacidophiles. Archaebacteria are also known as living fossils because they represented one of the earliest forms of life which experimented on the absorption of solar radiations for the first time, lived comfortably under anaerobic conditions, and developed techniques to oxidize the chemicals present in the substrate or the availability of oxygen. Methanogens. These Archaebacteria are strict anaerobes. Nutritionally, they are autotrophs, which obtain both energy and carbon from decomposition products. They occur in marshy areas where they convert formic acid and carbon dioxide into methane. This capability is commercially exploited in the production of methane and the fuel gas inside gobar gas plants, example Methanobacterium and Methanococcus. Some of the methanogen Archaebacteria live as symbionts, example Methanobacterium inside rumen of cow, buffalo, and heifer. The ruminants in fermentation of cellulose. Halophiles are named so because they usually occur in salt-rich substrata 2.5 to 5 m, like salt pans, salt beds, and salt marshes. Helicobacterium, they are aerobic chemotrophs. Their cell membranes have red carotenoid pigment for protection against harmful solar radiation. Under anaerobic conditions, halophiles cannot use external materials. At this time, they subsist on ATP synthesized by membrane pigmented system from solar radiations. Halophiles grown in salt pans and salt beds give offensive smell and undesirable pigmentation to the salt. Halophiles are able to live under high salt conditions due to the following reasons: presence of special lipids in cell membranes, occurrence of mucilage covering, absence of sap vacuoles, and hence plasmolysis, high internal salt content. Thermoacidophiles, these Archaebacteria have dual ability to tolerate high temperature as well as high acidity due to two reasons: branched chain lipids in the cell membranes and presence of special resistant enzymes capable of operating under acidic conditions. They often live in hot sulfur springs where the temperature may be as high as 80 degrees centigrade and pH as low as 2, for example thermoplasma and thermoproteus. Basically, these Archaebacteria are chemosynthetic, that is, they obtain energy for synthesis of food from oxidizing sulfur. Under aerobic conditions, they usually oxidize sulfur to sulfuric acid. 2H plus 2H2O plus 3O2 gives 2H2SO4. If the conditions are anaerobic, the Thermoacidophiles may reduce sulfur to H2S. Bicarbonates are also precipitated into the carbonate formed by their activity. Right? And we go on to mycoplasma, PPLO. Mycoplasma or molecules are the simplest and smallest of the free-living prokaryotes. They were discovered in pleural fluids of cattle suffering from pleural pneumonia by Knockhart and Roche, 1898. The organisms are often called MLOs, mycoplasma-like organisms, or PPLOs, pleural pneumonia-like organisms. The size ranges from 0.1 to 0.15 millimeters. Plasma membrane forms the outer boundary of the cell. A substantial amount of polysaccharides have been even acetylglucosamine oxide associated with cell membrane, which is rich in cholesterol. A cell wall is absent. Due to the absence of cell wall, the organisms can change their shape and are pleomorphic. Coccoid, Coccobacillus, helical, fine and branched or branched. Like other prokaryotes, mycoplasmas possess one envelope system. They lack organized nucleus, endoplasmic reticulum, plastids, mitochondria, Golgi bodies, lysosomes, centrioles, flagella, etc. The genetic material is presented is represented by a single DNA duplex, which is naked because of absence of histone association. The DNA duplex is not compacted as in other prokaryotes, but instead lies coiled throughout the cytoplasm. Ribosomes of 70S, granules of various types occurs here and there in the cytoplasm. Mesosomes absent, enzymes lie both freely in the cytoplasm as well as associated with the plasma membrane. DNA possesses a replicating disc at one end to assist in replication and separation of the genetic material. Some of them live as saprophytes, but majority parasites, plants and animals. The parasitic habit is due to the inability of most mycoplasmas. The parasitic habit is due to the inability of most mycoplasmas to synthesize the required growth factors, example Mycoplasma gallisepticum. Electron transport systems is rudimentary or absent. Reproduction occurs by fission or fist, forming a branching filament with numerous nuclear bodies, followed by constriction in between the nuclear bodies and separation of cells as new individuals. They mostly produce pleural pneumonia in domestic animals, atypical pneumonia and mycoplasmal arthritis, urethritis in humans, little leaf layer disease of brinjal, and witch's broom in plants. Mycoplasmas are not affected by penicillin inhibitor of wall formation, but are inhibited by tetracyclines. Cyanobacteria, blue-green algae. General factors are gram-negative prokaryotes, which perform oxygenic photosynthesis. Like plants. All right. And we have another line of to be completed. Mycoplasma is not affected by penicillin inhibitor wall formation, but are inhibited by tetracycline. We move on to Cyanobacteria, blue-green algae. The general factors. Cyanobacteria are gram-negative prokaryotes, which perform oxygenic photosynthesis like plants. They were the first organisms to make the atmospheric aerobic. The blue-green algae live virtually in all environments that contain water. The organism ranges from the Cyanobacterium are gram-negative prokaryotes, which perform oxygenic photosynthesis like plants. They were the first organisms to make the atmospheric aerobic. The blue-green algae live virtually in all environments that contain water. Organisms range from tropics to plains, and they occur in soil, freshwater, and ocean. In lakes and in the ocean, they form part of planktons. Some cyanobacteria live in icy waters at glaciers, others in hot springs where temperatures reach 85 degrees centigrade or more due to homopolar bonds. Some blue-green algae live as symbionts with other organisms, example lichens. Cyanobacteria can be unicellular. Example Spirullina colonial, example nostoc or filamentous, example Oscillatoria. Filaments contain one or more trichomes inside a mucilate sheet. Trichomes may be homocystous without heterocysts, example Oscillatoria, which shows apical oscillations, or heterocystous with heterocysts, example nostoc and anbana. Nitrogen-fixing nostoc, anabina live freely as well as symbionts in Azolla, Anthoceros, cycas roots, Gonera stems, nodules of Trifolium alexandrinitum. The cell wall possesses an outer sheath outside, which is jelly-like, slimy, and mucilaginous. The cell contents are divided into two regions: outer chromatoplasm, having photosynthetic pigments in free thylakoids, and inner colorless central plasm. The subwall is two-layered, whose inner wall is made up of peptidoglycan or mucopeptides built up from amino sugars, example glucosamine, and amino acids, example uramic and diaminopimalic. In cytoplasm, photosynthesis lamellae are present. These lamellae are thylakoids containing dominating pigments phyocyanin, allophycocyanin, and phycoerythrin, phycobilins in addition to chlorophyll A. Bacteria and blue-green algae lack mitochondria, true vacuoles, and endoplasmic reticulum. Sterols are absent in bacterium as well as cyanobacterium. True nucleus is absent. Two fibrils of DNA are either distributed throughout the cell or concentrated in the central part. Its chromosomes resemble bacterial chromosomes. Gas vacuoles are often present to regulate the buoyancy in water. The color ranges from green to deep purple, often blue-green. These colors are produced by different proportions of several pigments like chlorophyll A, carotene, xanthophyll, blue phycocyanin, and red phycoerythrin. Thylakoid membranes contain chlorophyll A, carotenes, and xanthophylls. Attached to thylakoids are minute structures called phycobilisomes. They have three types of water-soluble but protein-bound accessory pigments: phycocyanin, allophycocyanin, both blue, and phycoerythrin, red. Many forms show Guidicot phenomenon or chromatic adaptation, where color changes according to wavelength of light received by the cyanobacteria. Trichodesmium erythraeum is reddish-colored cyanobacterium which occurs in such abundance that a sea is named after its color, Red Sea. Red Sea contains Trichodesmium erythraeum. Nucleoid is in contact with a group of coiled membranes called the lamellosome. Lamellosome that develops from plasmalemma. Plasmids or additional small DNA rings may occur 70 years. Ribosomes are abundant. Cell inclusions are alpha granules or cyanophycean starch similar to glycogen, beta granules, lipid droplets, collagen granules, and polyhedral bodies. The reserve food is in the form of cyanophycean or mixophycean starch. Flagella are absent in vegetative as well as reproductive phase. Now, we have well-marked reproductive organs. Sexual reproduction and motile reproductive bodies are absent in cyanobacteria. Reproduction is very simple and takes place by vegetative means only. No trace of sexuality has ever been recorded by gene recombination. And can occur throughout conjugation, transformation, and transduction. The vegetative reproduction occurs by mere cell divisions. They reproduce asexually by binary fission in unicellular forms and fragmentation by breaking up regrowth of filaments. Heterocyst formations characterized by the presence of thick walls and yellowish contents. Each heterocyst is made up of two walls, an inner thin layer and outer thick gelatinous cover. Heterocysts develop from recently divided cells. It may be single or paired. On germination, it gives rise to a new filaments. Many filamentous forms asexual reproduction occurs by homogonia formation. They are identified by presence of biconcave discs or separation discs between two adjacent cells. The filaments get broken from such places into many homogonia, and each of them forms a new filament. Example, oscillatoria. Spore formation also occurs, exospores, endospores, actinides, and etc. Nitrogen fixation in cyanobacteria. Like many bacteria, several forms of blue-green algae have the capacity to fix atmospheric nitrogen into nitrogenous compounds. This capacity is restricted to filamentous heterocystous forms like nostoc, Anabaena, Autotrophic, etc. Under anaerobic conditions, some non-heterocystous forms can also fix atmospheric nitrogen, glucocapture, and oscillatoria. The additional capacity of nitrogen fixation along with carbon dioxide fixation makes them truly autotrophic plants. In this sense, they are considered to be largely responsible for the maintenance of soil fertility in tropical and temperate regions. Some species of blue-green algae have a great contribution to increase the fertility of rice fields in tropic countries like India. Example, Anabaena, Thallipoceros, and Autotrophic. Economic importance of cyanobacteria. The useful aspects of it are some 50 species of cyanobacteria are capable of fixing atmospheric nitrogen in soil. Example, Anabaena and Spirullum. Spirullum is being used as a source of protein-rich supplement to diet of humans. The balls of nostoc are used as food by Chinese and South Americans. All right. And on germination, it gives rise to new filaments. In many filamentous forms, asexual reproduction occurs by homogonia formation. They are identified by presence of biconcave discs or separation discs between two adjacent cells. The filaments gets broken down from such places into homogonia, and each of them forms a new filament. The spore formation also occurs causing exospores, endospores, and actinides. Now, the balls of nostoc are used as food by Chinese and South American. Food is called yoyucha. Cyanobacterium, like nostoc cytoma, are used for reclamation of yuja, sterile alkaline soil. In Sambo Lake of Rajasthan, Anabaena and Spirullum is produced in large numbers. Local people use it as green manure. Some species of Anabaena Thallipoceros help in conservation of soil, thus check in soil erosion. Few Cyanobacterium locate outside lichens, help in plant succession due to their growth on barren land. Oscillatoria is used too. This is pollution indicator. Harmful effects for forms like Anabaena not only spoil the taste of drinking water, but also produce toxic effects. Some cyanobacteria appear on buildings during rainy seasons and cause damage to bricks, etc. Skin infections may be caused by Cyanobacteria lynchbae. Toxin-secreting cyanobacteria are mainly responsible for water blooms. These on death emit foul smell. Water gets contaminated with heterotrophic bacteria and unfit for normal issue. So toxins secreted cyanobacteria are mainly responsible for water blooms. These on death emit foul smell. Water gets contaminated with heterotrophic bacteria and unfit for normal use. Some cyanobacteria like Vibrio release toxins which are harmful for aquatic fauna. So that's the end of that chapter there. There seems to be one or two pages I think are additional on viruses. So let me just read out that for you. In the chapter on viruses, they're basically broad-shaped viruses measuring 300 into 80 nanometers and have helical symmetry. have single-stranded DNA, which is 330 nm in length and have 7,300 nucleotides. Number of capsomeres in capsid is 2130. 5% RNA and 95% protein is present in TMV. Bacteriophages. Various infecting bacteria are known as bacteriophages or phages. Seawater can hold 100 million bacteriophages per microliter. Bacteriophages may be virulent or avirulent. Prophages are non-violent phages or non-infective phages are the phages which do not cause lysis of bacteria soon after their formation inside such bacterial cells which have prophages. Inside them are called lysogenic bacteria. Virulent phages or infective phages are the phages which cause lysis of bacteria cell at once. Most studied series of bacteriophages is T series, T2 or T4, T6, etc. The T-even phages are characterized by angular, bead, and concycled tail. Bacteriophages have a tadpole-like structure, that is with head and tail. Inside the head is present nucleic acid, generally DNA. Head is prism-like hexagonal, having length 950 angstrom and breadth 650 angstrom. Tail is also 950 angstrom in length. Joined to head by neck and collar. Tail is having hollow core of 80 angstrom and is surrounded by tail sheath. At the end of the tail, the end plate is present to which 6 tall tail fibers are attached and each is 1500 angstrom in length. Lytic cycle. T4 bacteriophage. The multiplication process of virulent phage is called lytic cycle. The main steps include adsorption, penetration, formation of new phages, and lysis. Bacteriophages attaches to the surface of bacterium by its tail fibers. Lysozyme creates a hole in the host cell. The tall sheath contracts and ejects the viral chromosomes DNA into the bacterium. Viral DNA controls the metabolic machinery of the host cell. It produces nucleases. Nucleases degrade DNA and mRNA of the host. Viral DNA is not affected as its cysteine bases are methylated. A number of copies of viral DNA are produced. Followed by synthesis of capsid proteins, polyamines, and lysozymes. The components assemble and form phage particles of viruses. The host cell ruptures to release viruses, lysis, which is called lysis. Lysogenic cycle or the the lambda phage. The multiplication process of temperate phage is called the lysogenic cycle. The phenomenon of existence of non-virulent prophage in the host cell is called lysogeny. The host cell in which the lysogeny occurs is called the lysogenic cell. Lysogenic cycle is shown by 1. The lambda phage, which also infects E. coli bacterium. Lytic bacteriophage multiply in host bacterium, which then undergoes complete lysis, degeneration to release the resulting daughter phages. The phage attaches to the surface of bacteria by means of tail, which produces a hole in a host cell wall and injects the phage DNA. The DNA phage produces a repressor, or C1, becomes non-virulent or temperate, and legs get integrated to bacterial chromosome at a specific site by means of enzyme integrase. The viral genome is now called prophage provirus. Prophage or provirus, it multiplies along with bacterial genome and is passed on to the progeny. Occasionally, the synthesis of repressor is stopped due to ultraviolet radiations or chemical factors. The temperate or non-virulent phage is now changed to lytic or virulent phage. The single-strand DNA of 174 or coliphage FD is known as plus strand. It forms the complementary or negative strand, the double strand of replicative DNA takes over the metabolic machinery of hosts to synthesize plus strands DNA and protein for assembly of new phages. The phagocytic reproduction. The whole virus enters the host cell except the envelope. It is quite common in RNA viruses. And which are two types, as RNA virus and RNA DNA virus. In RNA viruses, DNA has no role in their multiplication. After entering the host shell, the viruses produce enzyme replicase, which helps in producing more genetic RNA over the template. A copy DNA cDNA builds its complementary strand. The double-strand copy DNA attaches to host DNA chromosome and is now